Okay, we got it? Good.
All right. What we've got over here is a greatly enlarged view of the mitochondria. Up on top, this is the outer membrane of the mitochondria. We don't even need to show the whole thing. That's just mostly there to get things in and out of mitochondria. Here is the inner membrane of the mitochondria. Over here between the two membranes is that intermembrane space, the space between the inner and outer membranes. Now once again, the inner membrane of the mitochondria is intensely folded in these things we call cristae, so we can pack as much surface area of that membrane in as possible. The inner membrane is very protein rich. It's actually got about the same protein to lipid ratio as a typical hamburger, about 80% protein by weight, because it's packed full of all the enzymes and stuff needed for oxidative metabolism. Now over here is the mitochondrial matrix. Now let's see how this process goes. Oxidative metabolism depends on a series of large, gigantic enzyme complexes that span completely through the mitochondrial inner membrane. And these are often called the electron transport chain, or the respiratory chain. Okay, and what the electron transport chain, its name tells you what it does. It passes electrons from one place to another in a series of oxidation reduction reactions. Each time you oxidize something, you're going to release energy, and that energy will ultimately be used to make ATP. How we make ATP is a very bizarre thing, and we'll see that a little bit later. But the idea is the electron transport chain is going to take electrons from the coenzymes and ultimately pass them to oxygen. Okay, and we have some other goodies in there that we'll deal with later. So let's put three, in fact, I'm going to stick something over here too. We'll deal with this guy later. Okay, all right. We're going to have three large enzyme complexes. These are the electron transport chain enzymes. Each one of these enzyme complexes has about 15 to 20 separate proteins. They're huge. They're hundreds of thousands of molecular weight. They go completely through the membrane. Most of the enzymes in the electron transport chain have at their cores iron, often in the form of what we call a heme group. Okay, many of them have the iron-containing heme group that hemoglobin has. Others have iron ions that are complex with sulfur. Others just have an iron or two at the core. Now, what happens with iron? What happens when you combine iron with oxygen? You take iron metal, because yes, you rust it, you oxidize the iron. The oxygen takes the electrons from the iron, and now you turn your car into rust, especially if you live up in the Northeast. That's an exergonic process. It doesn't need to put any energy. Your car in the Northeast, where there's all that salt, especially in the Midwest where you put salt on the roads and stuff, your car will rust spontaneously on you. You don't have to put any energy. You have to put energy to get it all fixed up and a lot of money into it. But that's what's happening. So iron is really good at picking up or donating electrons. So in effect, all these iron ions in the cores of these things are used to pick up and pass down electrons. That's why we have, and these protein complexes are blood red in color because they have so much iron in it. Just like hemoglobin's red because of the iron in it. So what we're doing, and effectively the electron transport chain, we're effectively reversibly rusting these enzymes, passing electrons to these iron ions, and then passing them from the iron ions to other things, other proteins. So let's start. We're going to use NADH first. FADH follows a slightly different pattern. Now this first enzyme is often called NADH oxidase. That tells you something. What does it do? It oxidizes or removes electrons from NADH. So let's do that. We're going to take NADH, strip 2 electrons off of it, generating the positively charged NAD coenzyme. The NAD plus coenzyme, the oxidized version of this. And then we also take the hydrogen, we tear an electron from the coenzyme, we tear a second electron from the hydrogen, so now we have an H+. Now, 2 electrons are going to be passed into the electron transport chain, this first complex. They get passed around within the complex, from one protein to the next. Each time that happens, energy is going to be released. Now, what does that enzyme do with the energy? Well, it turns out that enzyme is capable of doing active transport of H+ ions. In other words, taking H+ ions from an area of low concentration and moving them to an area of high concentration. Of course, active transport is exergonic. It requires energy, or rather endergonic. It requires energy. Now, we talked about active transport before. We talked about co-transport. You let something in by facilitating diffusion and use that power to actively transport something else. Or just ATP hydrolysis. You use the energy of ATP hydrolysis to do active transport. In this case, this enzyme is going to use the energy released from these oxidation reactions to power the active transport of H+. So what we do is we take H+ and pass it through and pump it into our intermembrane space. And every time electrons go down this, more H+ gets pumped into that intermembrane space. Okay, we're done with our first enzyme. Now what we're going to do is transfer those electrons to our second enzyme. But what we're going to do is we're going to pass them, instead of going directly to this big enzyme complex, we're going to pass them to a little small hydrophobic molecule called a quinone, or ubiquinone is the name for that. It's often called coenzyme Q10. Have you ever heard of that? You get it at health food stores and stuff. Coenzyme Q10 is this little small hydrophobic organic molecule called ubiquinone. What it does is it picks up electrons from this first enzyme complex, and then it passes them to our second enzyme complex. So it acts like a molecular shuttle bus, carrying electrons from here to here. Now, why do we do that? Why not just have the 2 enzyme complexes directly pass electrons from one to the other? Well, the thing is, they're in the membrane. They have to move around in the membrane. Diffusion, in other words, moving around things, is strongly dependent on molecular weight. Now think of this as an analogy. Let's suppose you've got 2 big oil tankers. Let's suppose you're Exxon or something like that. One of them you have a drunk captain and he smashes it up into the ground in Alaska or something like that. No, okay. Well, anyway, at any rate, you got these 2 oil tankers. They're huge. They're 500,000 tons each. That's what big oil tankers weigh nowadays. Now let's suppose you want to do a crew transfer. You want to take some people from one tanker and take them to the next. What are you going to do? Are you going to have the 2 captains, especially one of them's Hazelton, are you going to have the 2 captains carefully steer these gigantic oil tankers so they're right next to each other, just barely touching each other, so the guys on one ship can jump across to the next ship? That'd be crazy. Instead, what you're going to do is you're going to put these guys in a small boat or in a helicopter or something. They can move from one tanker to the next easier than maneuvering these ponderous tankers right next to each other. Same thing here. It would be a lot quicker to use a little small molecule to transfer electrons from here to here, rather than directly passing them from one enzyme complex to another. So to go from one enzyme complex to the next, we're going to use these little small molecular shuttles. Okay, now we go to our second enzyme complex. Same thing's going to happen. We're going to pass them around within the complex, releasing energy each time we do that. Breaking the chalk again. This complex also is an active transport enzyme. it takes H plus, once again, from the matrix and actively transports it, once again, into our intermembrane space. So we've got two of these guys that are pumping H plus across the membrane into the intermembrane space. Now, once again, we're going to pass the enzymes again to another shuttle molecule, and that's what's called cytochrome C. It's a little small heme-containing protein. You guys are familiar with cytochrome C because that was a little small red protein we used when we did the gel filtration experiment. It's only about 12,000 molecular weight. It's actually not in the membrane. It's right next to the membrane. It sticks loosely to the polar parts of the membrane. But it acts as a shuttle bus. We transfer the electrons to cytochrome C and then to our third complex, which is often known as cytochrome oxidase. Guess what does it do? It oxidizes or removes electrons from cytochrome C. Once again, this complex passes the electrons around, releasing more energy. And once again, this complex also can actively transport H plus across the membrane using the energy of oxidation reactions. Okay, so each of these three enzyme complexes have pumped H plus across the membrane into the intermembrane space. And as we're going to see, that's really where the ATP production comes from. Not yet, we'll see that in a little bit. Okay, now, all good things must come to an end. We pass these electrons all the way from NADH all the way down to that tail end of the cytochrome oxidase enzyme complex. Now, we have to pass those electrons to a final resting ground, a final graveyard, a final electron acceptor. And what is that? It's the second most electronegative atom around. We're going to pass those electrons to oxygen. So, there's an enzyme in here that will split an oxygen molecule into an oxygen atom, two oxygen atoms. We're going to take this oxygen atom. We're going to also take 2H plus, and then we're going to pass 2 electrons. So we have an oxygen, 2H plus, 2 electrons, so we can add the electrons to each of the H plus, and then join it to the oxygen, and our final product, water. Okay. So what we're doing here is we're converting oxygen into water by adding H plus ions and electrons. In terms of energetic release, it's like taking hydrogen gas, which of course is H plus an electron on it, and oxygen gas, mixing together and throwing in a match. That's the same energetics here. And pound for pound, or gram for gram, hydrogen-oxygen reaction to make water is one of the most exergonic known. Hydrogen fluorine's a little better, and there's 1 or 2 other things. But who wants to breathe fluorine, right? I don't think it would be very good for us. Okay, but what's happening here? We're passing electrons from these coenzymes to oxygen. That's a tremendous drop in energy. And we're going to make water. So once again, we breathe oxygen in. We have 21% oxygen in our atmosphere, at least currently. What we breathe out is about 15% oxygen. We take about 1/4 of the oxygen with each breath out of our exhale. And then what we breathe out is saturated with water vapor. That's why when you go out on a cold day and you breathe out, you get this fog. Okay, because your breath is saturated with water vapor. That water vapor is largely produced during this process. We're basically turning oxygen into water, and we're getting a huge energetic boost in the process. Now, the downside of this whole thing, if you don't have oxygen, that means the electrons have no place to go. And pretty soon you get this backlog. Electrons get stuck at the proteins that they're at, shut down, get stuck back here, it gets stuck back here, and finally you just shut the entire electron transport chain down. It will not work without oxygen as a final electron accept. And what that means, if you shut that down, ultimately you're going to lose 90+% of your ATP production. Very bad indeed. Now the other thing is there are certain types of molecules that can strongly inhibit, especially that third enzyme, cytochrome oxidase. For instance, cyanide. Cyanide binds to and shuts this baby down. Azide, that's what we use as a bacterial growth inhibitor, does the same thing. It's also as toxic as cyanide. By the way, cyanide, a couple milligrams of cyanide or azide, and you will feel it. You will feel short of breath. It's not pleasant. A couple, 100 or 200 milligrams, you're going to be either in the ER in the hospital or in a zippered plastic bag. But 200 milligrams probably less. This stuff is so toxic because it specifically shuts down cytochrome oxidase, shuts off the whole electron transport chain. That's why it was used in things like Jim Jones Kool-Aid back in Guyana. Okay, that was Lancet. That's what happens with religious extremists. And that's why the Nazis used it. It was Zyklon B that was used during the Holocaust. It was a pesticide. You would fumigate things to kill off rats, roaches, and stuff like that in houses. And then the Nazis decided it's a good way of killing off large numbers of Jews efficiently, so they made huge-scale production of it, and that was the gas chambers in the Holocaust that killed 6 or so million people. It was quick and efficient way of mass murder. So cyanide has a really nasty reputation, especially when you mix it with Germans. Okay, yes.
That first thing that you said, the F as a shuttle bus,
Yeah.
what was it called?
That's called ubiquinone or coenzyme Q10. You go to a health food store, you get these supplements, vitamin supplements, coenzyme Q10, and it will say it helps metabolism. The reason it does is because you need it to transport things. And it's a little small, about 500 molecular weight, hydrophobic organic molecule, so it lives in the membrane. Okay? All right.
So that's supposed to be like a Q?
Yeah, Right. Yeah, that's a Q. Oh, I know. I worked handwriting in elementary school all the time. It hasn't changed.
I'm chicken scratch.
Yeah, I know. Yeah, even my wife can't always read my writing. It's scary though. Okay. All right. Now, interesting thing here. We've taken NADH, taken electrons, passed them all the way to oxygen here, right? Okay. We got 3 enzyme complexes. Each one of them pumps H+ from the matrix to the intermembrane space. And one NADH is able to produce maybe 3 ATPs? Is there a connection here? Maybe. Now, something else. So far we haven't looked at FADH because FADH uses a slightly different path. What happens here with FADH is an enzyme takes electrons from it, and then it passes those electrons to our second enzyme complex. It bypasses the NADH oxidase entirely. So we go straight from here to this enzyme complex 2 and 3. So we got 2 enzyme complexes that pump H+, and guess what? FADH makes 2 ATP. So there is a connection. Okay. All right. So now we have run the electron transport chain. So far what we've done is produce water from oxygen, used up oxygen, found a good place to get rid of those electrons, and pumped a heck of a lot of H+ across the membrane into the intermembrane space. Now we still haven't done one thing that's supposed to be really important in this process, the whole idea behind this process. How do we make ATP? We haven't even touched ATP on this yet. Yeah, you're shaking. Yeah, where's the ATP? Where's the beef? Where's the ATP? Okay, we're going to find out in just a moment, folks. All right.